Welcome back. Physics builds models. Chemistry builds structures. Biology builds evolutions. 3 ladders, 1 way of thinking. And today, we drop that thinking straight onto a real exam idea, the 2018 VCE Biology NHT MCQ set on regulation of biochemical pathways. Today's focus: enzyme-substrate interactions, competitive inhibition, reversible binding, and hormone-driven metabolic changes across temperature. Identification of key components. Molecule Y, enzyme, the large gray structure. It acts as a biological catalyst. Notice that molecule Y remains completely unchanged from the start to the end of the reaction. Structure A, active site, the specific 3D pocket or groove on molecule Y. Its shape is complementary to the shape of molecule X, allowing specific binding. Molecule X, substrate, the black reactant molecule that binds to the active site. Molecule Z and W, products, the 2 smaller black fragments released at the end of the reaction after molecule X is broken down. The 3 stages of the reaction. Left panel, initial state. Substrate, molecule X, approaches the enzyme, molecule Y. The active site, structure A, is vacant and ready for binding. Middle panel, enzyme-substrate complex. The substrate binds to the active site to form an enzyme-substrate (ES) complex. Within this complex, bonds within molecule X are stressed or broken, lowering the activation energy of the reaction. Right panel, product release and enzyme recovery. The reaction completes, splitting molecule X into 2 smaller products, molecules Z and W. The products leave the active site, while molecule Y, the enzyme, and structure A, the active site, remain unaltered, free to process another molecule X. A is incorrect. Molecule Y is the enzyme, not the substrate. B is incorrect. Molecule X is the substrate, not the enzyme. C is correct. Structure A is the binding region on the enzyme, which is the active site. D is incorrect. Molecule Z is a product formed from the breakdown of molecule X, not a reactant. Diagram analysis and key concept. Competitive binding. The diagram shows molecule P binding directly to a portion of structure A, the active site, of molecule Y, the enzyme. This directly blocks the substrate, molecule X, from occupying the active site. Reversibility evidence. The question states that increasing the concentration of molecule X increases the rate of production. In enzyme kinetics, if increasing the substrate concentration, substrate can outcompete the inhibitor and restore the reaction rate, the inhibitor must be reversible. An irreversible inhibitor would permanently occupy or destroy the active sites regardless of how much substrate is added. Option breakdown. A is incorrect. Molecule P binds to the active site of the enzyme, not to the substrate, molecule X. Molecule X does not change its chemical structure or shape due to P. B is correct. Because the inhibitory effect of molecule P can be overcome by increasing the concentration of substrate, molecule X, molecule P is a reversible competitive inhibitor. C is incorrect. Denaturation involves a permanent conformational change, unfolding, of the enzyme's tertiary structure, which cannot be reversed or overcome simply by adding more substrate. D is incorrect. Inhibitors can be various chemical species, often small organic molecules, ions, or peptide chains, there is no evidence or basis to conclude molecule P is a nucleic acid made of nucleotides. An experiment was conducted to measure the effect of a hormone on oxygen consumption in liver cells at different temperatures. 2 liver cell samples were prepared in the following way. Sample A, 10 g of liver cells in a nutrient solution. Sample B, 10 g of liver cells in a hormone and nutrient solution. The results of the experiment are shown in the table below. A is incorrect, as temperature increases from 10 degrees Celsius to 30 degrees Celsius. The absolute increase in oxygen consumption due to the hormone actually increases. The difference between sample B and sample A goes from 8.04.1 equals 3.9 to 21.0 10.1 equals 10.9. B is incorrect. Enzymes undergo inactivation, slower kinetic energy at collisions, at lower temperatures, not denaturation. Denaturation occurs at high temperatures, above 30 to 40 degrees Celsius. C is incorrect. From 30 degrees Celsius to 50 degrees Celsius, oxygen consumption decreases dramatically in both samples, from 10.1 down to 1.5 in sample A, and 21.0 down to 3.1 in sample B. Comparing sample B, hormone plus nutrient, with sample A, nutrient only, across all temperatures. 10 degrees Celsius, 8.0 divided by 4.1 almost equals 1.95, nearly double. 20 degrees Celsius, 15.5 divided by 7.8 almost equals 1.99, nearly double. 30 degrees Celsius, 21.0 divided by 10.1 almost equals 2.08, nearly double. 40 degrees Celsius, 13.4 divided by 6.7 is equal to 2.00, exactly double. 50 degrees Celsius, 3.1 divided by 1.5 almost equals 2.07, nearly double. The correct option is D. The liver cells in the hormone and nutrient solution consume approximately double the amount of oxygen that is consumed by the liver cells in the nutrient solution. Enzymes control biochemical pathways through precise interactions between their active sites and specific substrates. In the diagram, structure A functions as the active site, the region where molecule Y, the substrate, binds to molecule X, the enzyme, forming an enzyme-substrate complex that produces molecule Z and molecule W. This regulation can be disrupted by competitive inhibitors. Molecule P binds to the same region of the active site as the substrate, temporarily blocking access. Because increasing substrate concentration restores product formation, molecule P acts as a reversible competitive inhibitor. Temperature also influences pathway regulation. In liver cells, the presence of a hormone consistently doubles oxygen consumption across temperatures, with peak activity at 30 degrees Celsius. Beyond this point, oxygen use declines as enzymes begin to denature, reducing the hormone's effectiveness. At the end of every lesson, I want to leave you with one idea: learning becomes powerful when you climb the logic ladder. Physics moves from situations to models to equations. Chemistry builds from structure to property to reaction. Biology grows from variation to adaptation to survival. Different subjects, but the same underlying logic. When you learn to decode systems, build ladders, and construct logic, you don't just solve problems, you understand how they fit together. If you'd like to keep climbing this ladder with me, you can find more lessons, breakdowns, and reconstructions on my channel. Thanks for learning with me today.